Welcome back. In today's class, we are going to learn about sea sponges. How these sea sponges are in symbiotic relationship with lower organisms. How they benefit other organisms and how they get mutually benefited. These sea sponges are the colorful part of the marine ecosystem, which is seen in the ocean beds. They are in association with coral reefs, as we learned in the previous session. These sea sponges are seen to be in symbiotic association with various green algae, primitive algae, or also with cyanobacteria, that is the blue-green algae they are in symbiotic association with. These sea sponges are generally called as the filter feeders. As I name it, it is sea sponges. As the name depicts, it is highly porous in nature. The water which passes through it gets filtered. The organism that is there in the water or the mineral nutrients that is there in the water are metabolized by these sponges. That is why they got this name, filter feeders. They are in the aquatic environment in the marine ecosystem, believed to be in symbiotic association with the primitive algae, especially with cyanobacteria, which is the blue-green algae, freshwater living or marine area living, generally seen attached to a hard substratum. That's why they are seen in the rocky marine oceans and also seen in the ocean beds. They are never free floating. They also act as a habitat for primitive organisms to reside on, but it also utilizes or consumes the minute organism that passes through it. This also acts as a fish feed in the natural marine ecosystem. This is the ultra structure of the sea sponge, which is hollow in between, long, elongated, tubular in nature. It has spicules, it has osculum, and it has several constituents which makes the sea sponge structure complete. So to learn that, we need to decode each type of subunit that is there. Mainly we will be focusing on this amoebozoite and canocytes. This is what we will be focusing the most. Symbiotic association of this sea sponges is seen to be in very good symbiotic relationship with amoebozoites. These amoebozoites and canocytes together brings mineral nutrients to the sea sponges, and it gives it in the utilizable form to the sea sponges, where amoebozoites and canocytes together do this. And this sea sponge in return gives a perfect habitat or a perfect shelter for these two organisms to reside on. As I say, the structure or the consistency of the sea sponges makes them a perfect habitat for the other organism to live. Like this, sea sponges are generally considered carnivorous in nature, as they consume the organisms that comes through this. Sea sponges need not be very small, need not be attached to the substrate at the ocean beds, but it is also seen in the rocky environments, which can look. This is called as the giant barrel sea sponge. This is the giant barrel species sponge. Giant is because of its size and the barrel shape. That's why it is called as a giant barrel sponge. And this also looks very presentable like this. It will be closed generally, but it senses any prey when it approaches it. It opens like this, looks very presentable. When the organism comes and sits on it, it closes and starts utilizing it. There are many places or particular part in which they reside, the symbiotic organisms. Sometimes in the external surface or the external layer of the sponge, that is called as extracellular exasymbiotic. Sometimes it is seen adhered to the external structure, but the metabolites are sent inside the sea sponges. That condition we call the symbiosis as extracellular endosymbiosis. And also at times the organism enters inside the sea sponges and performs the symbiotic activity. Then that is called as intercellular symbiosis. Sometimes the organism enters the nuclei of the sponges. That is called as internuclear symbiosis. So the position at which these photosynthetic organism resides matters a lot based on the specific function they perform. These are the list of sea sponges in symbiotic association with list of microorganisms. It's not just sea sponges relationship with algae. It also has good relationship with the bacteria and microorganisms in general, which also includes fungi. It also includes certain protozoans. So these are the summarizing list. So to speak about the symbiotic association, I was telling about the important two members that we will be learning. One is amoebozoites and the other one is canocytes. These amoebozoites are motile amoeba-like structure, which is meant to transfer the metabolites or mineral nutrients from canocytes to the sea sponges. That is the duty of this amoebozoites, which is meant to be like a mobile organism or a mobile interface between the canocytes and the sea sponges. These amoebozoites are amoeba-like structures, motile in nature, capable of helping the sea sponges to eat, to grow, and also to reproduce. To speak about the amoebozoid subunits, they are generally classified into various forms based on the activity they perform. Amoebozoids, the minute we say amoebozoids, they help in to eat, they help in to grow, they help in to reproduce, which means they are available in various forms. This amoebozoids also help the sea sponges to make its structural part. It acts as a structural constituent, helps in the building blocks of the sponges to have its morphology appropriate. For that, sclerocytes are used, where these are the specialized amoebozoids which secretes biosilica, which acts as a building block or which acts as the constituent such as calcium and biosilica produces makes sure the sponge structure is having good tensile strength and rigidity. The rigidity of the sponge structure, the flexibility or the tensile strength of the sponge structure is decided by the sclerocytes, because they are the ones which produces enough of amount of calcium to the sea sponges so that its structure can be maintained strong, so that in turn these amoebozoids can reside there. Archaeocytes are the ones which produces galactid. This is a glue-like, moist, jelly-like substance secreted by the special type of amoebozoids, which helps in holding the spicules together, or all the part of the sea sponges together. Spicules are nothing but the external infertile hair-like substances protruding out of the hollow pore of the sponges. Then lymphocytes are nothing but the collagen fibrils are secreted. Lymphocytes are produced by special type of amoebozoids which produces collagen fibrils. These collagen fibrils allows the endoskeleton to be flexible and also at the same time to be rigid to avoid breakage. These are the this is the ultra structure of the sea sponge, where there are panocytes. They are nothing but the ones pinacocytes. These are the ones which helps in the maintaining of the structure of the sea sponges. The structure of the sea sponges are maintained by them. And there are mesenchymes, which are the free living minute organisms. And then this is the canocytes that I was emphasizing on, that we will be looking into. Canocytes are the ones which, if this is the sea sponges, this is the amoebocytes, this is the canocyte, and to which the amoebozoid is connected with. When we take a transverse section, these blue color are the ones which are the amoebozoids. This purple color ones are the ones which are canocytes. These canocytes are protruding in nature. It has a flagella and it has a collagen liberation from it, which is performing a specialized metabolism called as phagocytosis to engulf the mineral nutrients. When we take this is the sea sponge, this is the hollow structure, osculum is the open pore to which the spicules are attached. When we take one subunit and when we try to elongate, we can see the symbiotic relationship of the canocyte and amoebozoids. These canocytes are being having a capability to entrap or engulf the mineral nutrients, is done by the canocytes, metabolized and lysed and given to amoebozoids. These amoebozoids are the mobile messengers or the mobile transporters which can transport mineral nutrients and made available to the sponges. So this is how it works. We have canocytes which traps the mineral nutrients and metabolizes it. Amoebozoids helps in transporting it to the sea sponges, and the sea sponges are giving in turn the shelter to these canocytes and amoebozoids. This sort of a symbiotic relationship between canocytes and amoebozoids are very essential to reside there. Only one person can not serve the purpose, is because canocytes are immobile in nature. Amoebozoids cannot perform phagocytosis. So together they perform phagocytosis, they metabolize them, make it in the form that can be readily utilized by the sea sponges, and then that is sent to amoebozoids, which transports it to the several part of the sea sponges. So this is the symbiotic relationship seen by the sea sponges. To speak about its reproduction part, is they undergo sexual and asexual reproduction. Asexual reproduction is by fission, where the thalli gets divided into small bits and pieces, where each bit now develops into a new individual. Fragmentation, again, yes, happens that way. Fission need not be binary fission, it can be numerous, and the budding is, the small part of the thalli gets swollen and then matures and then detaches from the parent thalli and develops into a new individual. To speak about the sexual reproduction of the thalli, the sea sponges produces mature anthriozoids or mature sperm cells. These adult sponges produces mature sperm cells, which travels by the water current, goes and fertilizes the fresh sea sponge, which carries ova in it, or which carries egg in it, and then that egg cell is been fertilized. There is only one egg cell produced, but there are numerous sperms that are produced. So these numerous sperms goes and attacks the egg cell, where the fittest one fertilizes the egg cell and the zygote is produced, larvae is produced. Once the larvae matures inside the sea sponge, gets released into the water current, goes and falls onto the suitable substratum, on set of favorable condition, develops into a new individual, and then that undergoes a certain development, and then that produces sperms or produces egg, gets fertilized, or goes and fertilizes another sponge, and then the cycle keeps repeating. So this is all we have for today's class. So from where did we start? We spoke about what exactly are sea sponges, what are its symbiotic association with, what is its ultrastructure. It is in symbiotic association, especially two members that we were focusing, amoebozoids and canocytes. These amoebozoites and canocytes have their various versions in which they exist. There are various ways in which they help sea sponges. It is not just delivering the food, it is not just metabolizing the food for sea sponges, but also maintaining the ultra structure of the sea sponges are contributed by amoebozoites. Canocytes in relationship with amoebozoites are very beneficial in performing phagocytosis, and then they in turn accommodate inside the sea sponges, which is a perfect habitat for them to live. They perform sea sponges perform sexual and asexual reproduction by fission, fragmentation, and budding. Asexual reproduction is seen. Sexual reproduction is by the production of sperm cells, and the egg cell after fertilization produces larvae which falls onto the suitable substratum and develops into a new individual. So that is all we have for today's class, and all the very best. Thank you for watching this video. See you in the next video. Thank you.